Everyone, in this video we will be discussing regarding the extra embryonic membranes. Why the animal requires these extra embryonic membranes. So these extra embryonic membranes are being seen in the development of the reptiles, aves, mammals. So what is the function of these extra embryonic membranes? So these extra embryonic membranes which are being present in the developmental stage, they are being responsible for the protection of the embryo, or these give the nourishment to that of the embryo, or they act as a kidney where the filtration of the blood takes place, or it may store even the nitrogenous waste. So for various of the reasons, these extra embryonic membranes are being seen in the classes of reptilia, aves as well as mammals. Now the animals which bear or which have these extra embryonic membranes are called as amniotes. They are called amniotes. And the animals or the classes of the phylum Chordata which do not contain these extra embryonic membranes are said to be anamniotes. So example for the anamniotes are the two classes. One is the fishes or pisces, the other one is called as amphibians. So they are said to be anamniotes, and reptiles, aves and mammals are said to be amniotes. Now, in case of the human beings, the four extra embryonic membranes which are present in the developmental stages, so they are chorion, amnion, yolk sac and allantois. So how these yolk sac or allantois, amnion, chorion, how they are being formed? So during the developmental stages, once after the stages like blastulation, gastrulation, neurulation occurs, thereafter will be the formation of these extra embryonic membranes. Now, if you look after the diagram here, so in the first diagram, it depicts about the embryo which is their initial stages, where the entire, you can see that the outer layer is said to be the trophoblast which is present, and from this trophoblast, so there will be formation of the cells which are said to be amniogenic cells. And here, in case of the embryonic development, there are presence of the two layers, namely the ectoderm and endoderm, which together form a bilaminar embryonic disc, and in the later they will get converted into a trilaminar embryonic disc, which generally consists of the three germ layers. First one is the endoderm, next is the ectoderm, and the third one is mesoderm. So here the cells which are involved in the formation of this trilaminar embryonic disc, so they are, one is called as the epiblast cells and the other one is called as the hypoblast cells. Now in between, if you see, in between the amniogenic cell and the epiblast cell, so there is presence of this cavity, so that is called as amniotic cavity, and this amniotic cavity consists of the amniotic fluid, whereas on the lower side the hypoblast cells which are present, so these hypoblast cells later form the endoderm, and later they this particular pouch or the blastocele which will be present, so that gets converted into a primary yolk sac, and this primary yolk sac in the later developmental stages will become the secondary yolk sac and finally forms the yolk sac. So that is in case of the first diagram. So apart from that of this particular embryonic stage, surrounding you can see there is a presence of the extra embryonic, the mesoderm. So why we call this as extra embryonic mesoderm? Because the mesoderm which is present, so that is formed in between the endoderm and ectoderm. That is the reason we call it as extra embryonic mesoderm. Now in the next stage, what you can see here is the amniotic cavity is being present, along with that of the presence of the yolk sac. So here the yolk sac is said to be the primary yolk sac, whereas here it is said to be the secondary yolk sac. Why it is so? That is because you can see this mesoderm which is present. So this mesoderm, just below to that of the mesoderm, there will be a formation of a large cavity. So this large cavity is called as extra embryonic coelom. Means the coelom is being formed, and surrounding this coelom there will be presence of the mesoderm. So the mesoderm which is there, which is being connected with that of the ectoderm. Okay, mesoderm and ectoderm. So that is called as somatopleure. The layer where the mesoderm is being connected with that of ectoderm. So that is called as somatopleure. And towards here you can see that the mesoderm which is being connected with that of the endoderm. So that is called as splanchnoplure. That is called as splanchnoplure. So these are the two important points for your competitive exams. What you have to remember is the mesoderm with ectoderm forms somatopleure, and the mesoderm with endoderm forms splanchnoplure. So here the splanchnoplure will be seen, which is being connected with the endodermal region, and this outer one which is surrounding to that of the coelom, so that is said to be somatopleure. So as the coelom is being covered by that of the mesodermal lining, the human beings are said to be eucoelomate, because generally according to the definition we say that the body cavity which is being lined by the mesoderm, so that is called as eucoelomate. Okay, so this is how the coelom will be formed. Next is, in the later stages, what you can find is there is a formation of the 4 extra embryonic membranes, namely yolk sac, allantois, amnion and chorion. So let us get into the details of these 4 extra embryonic membranes, what is their function and how they are being formed. So among the 4, the first one is said to be the yolk sac. So yolk sac is a small pouch-like structure which is being present below the embryo. And this yolk sac you can see, so this yolk sac is being derived endoderm inside and mesoderm outside. So these are very, very important for your various of the competitive exams. Yolk sac is being derived from endoderm inside and mesoderm outside. Okay, so that is yolk sac. The function of this yolk sac is it helps in the nourishment of the embryo. So that is commonly seen in case of reptiles and aves. But in case of the human beings, the yolk sac does not provide any nourishment to that of the embryo. Why? Because this functioning of transportation of the nutrients is being done by the placenta. Okay. Henceforth, as it is being involved in delivering the nutrients, it is said to be extra embryonic gut. But in human beings, as it lacks, as it does not provide any nutrients to that of the embryo, here associated with the digestive system, yolk sac is said to be vestigial. Then, if it is vestigial, then what is the use of this yolk sac? Yolk sac in human beings is being involved in a process called as hematopoiesis, where hematopoiesis refers to formation of the blood, and yolk sac is also involved in the erythropoiesis, that is formation of the blood cells, basically that is in the process of the formation of the blood. Now the yolk sac continuously produces or synthesizes the blood cell or the blood up to 7th week, and later this yolk sac, you can see the size here, this is primary yolk sac, here it is the secondary yolk sac. So comparatively the size of the yolk sac starts diminishing during the developmental stages, as it does not have a role in providing the nourishment to the young ones. So this yolk sac, which is being confined to the hematopoiesis, so it continues the production of the blood up to the 7th week, and later it completely diminishes in the size, and there is a formation of another organ that is called as liver. Okay, so liver thereafter takes part in that of the hematopoiesis. So this yolk sac is being located below the embryo. Is that clear? So this is the first extra embryonic membrane, yolk sac. Second, next is allantois. Allantois is another structure or extra embryonic membrane which is also present below the embryo, and this allantois is a hollow sac-like structure which is being again formed or derived from endoderm inside and mesoderm outside, as same as the yolk sac. So both the structures have endoderm inside and mesoderm outside. Now this allantois, in case of the reptiles as well as in the aves, allantois is responsible for storage of the nitrogenous or metabolic waste. So there it is said to be extra embryonic kidney, because the kidney does the same function. But here also allantois is not at all involved in that of storage of the waste in humans, because that function is also done by the placenta. Then what is the function of this allantois? Allantois in human beings is responsible for the formation of the blood vessels. So what those blood vessels are which are being derived from allantois? So they are said to be umbilical arteries and umbilical veins. So these umbilical arteries and veins are being formed from allantois. So that is the main function of the allantois. Okay, and later these arteries and veins are being present in that of the umbilical cord, where this umbilical cord connects the developing young ones with that of the mother. So that is allantois. Okay, so thereafter, after the second extra embryonic membrane, the next extra embryonic membrane which is present, that is called as amnion. So amnion, plays a very vital role, and this amnion consists of a large cavity. You can see here, this amnion is being present above to that of the embryo, and this amnion is consisting of a cavity called as amniotic cavity, and this amniotic cavity is filled with amniotic fluid. And here this amnion is being derived, very important, this is being derived from, this is the entire, that is ectoderm, and surrounding the ectoderm is the presence of mesoderm. So which means amnion is derived from somatic mesoderm or mesoderm outside, and ectoderm inside. Very important. Amnion is formed from or derived from ectoderm inside and mesoderm outside. Now this amnion, which consists of the amniotic fluid, so this amniotic fluid has many number of the function, where one of the function is amniotic fluid acts as a cushion and it protects the embryo against the mechanical injuries or physical injuries. So that is first function. Second function is amniotic fluid allows the fetal movement. This is embryo. Later in the developmental stages, the fetus start making its movements inside that of this amniotic fluid, and thereby it helps in the muscular movements also. That is second point. Third point is this amniotic fluid prevents attachment of the embryo to that of the amnion. So that is third function. Fourth function is this amniotic fluid maintains homeostasis as well as the electrolytes. Next is amniotic fluid allows, it allows the growth and development of the lung. Okay, so that is also being done by that of the amniotic fluid. Now during the developmental stages, what happens is this amniotic fluid consists of the embryonic cells. So during a procedure or a technique, we call it as amniocentesis. So a microliter of this amniotic fluid is being extracted and that it is being analyzed. So when it is being analyzed, as this amniotic fluid contains the cells of that of the embryo, so one can easily determine the sex of the young one or the fetus. So that entire technique is called as amniocentesis, which is generally banned in India. So these are the various of the functions which are being of the extraembryonic membrane called as amnion. And the last extraembryonic membrane is said to be chorion. So chorion is said to be the outermost layer that is formed or derived from mesoderm inside and ectoderm outside. Very important, you should remember, in the amnion, mesoderm is formed or it is present outside, ectoderm is inside. But in case of chorion, ectoderm is outside and mesoderm is inside. So that is chorion. Now the function of the chorion is, chorion generally in case of reptiles as well as aves. So this chorion helps in the exchange of the gases. Thereby it acts as an extra embryonic lung in reptiles as aves. But in case of the human beings, this chorion does not facilitate any respiration process. Okay? Why? Because the functioning of the exchange of the gases is being done by the placenta. So thereby in human beings it does not have any role in respiration. Then what is its function? So this chorion, okay, it contains a cavity. So that cavity consists of the chorionic fluid. So this chorionic fluid acts as a shock absorber and protects the embryo. That is first function. The another function is this entire chorion, okay, gives rise to many number of these finger-like structures. So they are called as chorionic villi. And these chorionic villi later gets attached with that of the maternal blood vessels and forms the placenta. Henceforth, these chorionic villi, as they will form the placenta, so these are said to be placental chorionic villi. Remember, these are the four membranes which are yolk sac, allantois, amnion and chorion. So these are said to be extra embryonic membrane, which means they are not inside the embryo, but they are present outside the embryo. And these extra embryonic membrane, either they may protect or they may provide the movement of the embryo, or they may help in the formation of the arteries and veins as well as the blood. So this is all about the extra embryonic membranes.